my name is Blake, and today we'll be talking about the unicellular organism I've chosen, which is Pelagibacter ubique. And I will be talking about the discovery, the cell shape, genome, organelles, reproduction, and the metabolism of this organism. Pelagibacter ubique is found in the domain bacteria, and it is a part of the HTCC1062 strain, and in the SAR11 clade. These organisms are some of the smallest and simplest self-replicating and free-living cells, which are heterotrophic alpha proteobacteria. It was originally found off the Oregon coast and cultured in the Giovannoni Laboratory in 1990. The Giovannoni Laboratory was a part of the Oregon State University. They are found in coastal surface waters of temperate and polar oceans. They are known as an oligotrophic marine bacteria, which means that they live in a low-nutrient ecosystem. It was first cultured in 2002, and Candidatus Pelagibacter ubique was the first cultivated member of the SAR11 clade. These are among the smallest free-living cells and make up 25% of all microbial plankton cells. Pelagibacter ubique has two phases in which they can enter. These are the log phase and the stationary phase. Cells that are in the log phase are log or crescent shaped. The length of these cells vary from 400 nanometers to 1300 nanometers, and the diameter of these cells range from 190 nanometers to 250 nanometers. It has an inner and outer membrane with a thin periplasmic space separating them. Inside the cell, you will find a nucleoid, ribosome, and cytoplasm. You will find the nucleoid on the convex side of the cell and cytoplasm with the ribosomes on the concave side of the cell. These cells keep their shape by using IF proteins. They will divide by fission when they are approximately double in size. On this picture, in A and B, you will see them in log phase. You can see where the nucleoid and the ribosomes are located, including the periplasmic space, which is this, or the white here. And the nucleoids are on the convex side, and the ribosomes are on the concave side. The outer membrane is the dark blue section. The inner membrane is the light blue, the periplasmic space, and the peptidoglycan layer is the white, and the cytoplasm is orange. Cells that enter stationary phase are more spherically shaped. These cells vary in length from 600 nanometers to 900 nanometers. This has one exception, which is that when a cell fails to complete cell division, but it continues to grow. This phase has different subcellular components, which are that the cells are wider, the cytoplasm is smaller, and the volume of the periplasm is increased. Pictures C and D show it during stationary phase. So your ribosomes are on this side, the nucleoid is on that side, and as you can see, the periplasmic space is obviously larger as the cytoplasm is increasingly smaller. P-ubique cells, as well as being the smallest free-living cell, has one of the smallest genomes that we have discovered. It is one of the smallest genomes found in autonomously replicating cells, which could be as a result of the genome streamlining. Its genome has adapted to an ecosystem in which nutrient concentrations are near the limits of where its transport systems can still function adequately. Their genomes have also evolved to execute only essential functions for life. It also has the presence of DNA uptake and competence genes, which suggests that this organism has the ability to acquire foreign DNA. Within these cells is a nucleoid, ribosomes, and the cytoplasm. The nucleoid is located on the convex side of the cell and is essentially located within the cytoplasm. So it's just along the convex side here. And the nucleoid can be identified by its fibrous texture and lack of ribosome-like particles. It is also spatially segregated from the ribosomes on opposite side of the cell. The ribosomes are found on the concave side of the cell here, and are peripherally located within the cytoplasm. They have a high concentration of 10,000 to 12,000 ribosomes per micrometer cubed. They also have a relatively high abundance in the cytoplasm relative to the slow growth rate of this cell type. The cytoplasm is half occupied by the centrally located nucleoid. Its volume is reduced in stationary phase. There is also the periplasmic space, which I mentioned is this here and this. And this is located in between the inner and the outer membrane. It has a thinner diameter along the cell body of the organism and a larger at the poles. So it's thicker here than it is here. And there are one or two layers, which is the peptidoglycan layer and scattered particles. The periplasm matrix is more translucent than the cytoplasm. It can occupy 20 to 50% of the cell during log phase and increases to roughly 50 to 70% during stationary phase. There is also an outer membrane pore complex that have pore-forming channels that are found in the outer membrane. Pelagibacter ubique is a bacteria that reproduces asexually by using the division process of fission. Although it is uncommon, they can also undergo asymmetric division. Short cells that have little curvature are presumably products of recent cell division. They have long filaments that are type 4 pili found in dividing cells. These filaments extend from the cell division site into the extracellular matrix. The pili are not involved in motility, but they play a role during cell division. They use pili to attach substrates in the ocean and help with the completion of fission, or they allow the daughter cells to float away after division to ensure low local concentrations of cells competing for the same resources. This organism is not competitive in dilute ecosystems. This is because they waste energy producing metabolic enzymes that will never process substrates that are close to their turnover capacity. It is suggested that these organisms are partially dependent on other community members for basic metabolic building blocks. They are adapted for low nutrient environments, which is why they are classified as an oligotrophic marine bacteria. Oligotrophic is characterized by a low accumulation of dissolved salts supporting a sparse growth of algae and other organisms, and having a high oxygen content owing to the low organic content. It encodes the basic functions of alpha proteobacterial cells, which enables them to grow in low nutrient ocean systems. It is shown to metabolize pyruvate as a carbon source and used as a positive control, and it also plays a major role in the carbon cycle. P. ubique is devoted to the C1 metabolism that produces energy but not biomass. Their metabolic pathways and transport systems react different in light and dark. Under dark conditions, amino acids are transported, and under light there is an upregulation of genes involved in glycolysis. The periplasmic space has additional particles to the peptidoglycan layer, which suggests that large proportions of resources to enzymes that modify and transport nutrients, such as ABC transporters. So that was my presentation, and I hope you learned something today. Thank you for listening.